Hello, this is section 17.4 of section 4 of chapter 17 on nucleic acids. The topic for today is going to be RNA, or for this segment is going to be RNA and transcription. And we've already talked about the structure of DNA and DNA replication, the double helix, structures of nucleic acids in general. Now we're going deeper into this. Excuse me. We're going to be talking about the role of ribonucleic acid in protein synthesis. You see here the picture on this slide is a typical transfer RNA molecule. It has it's the smallest of the nucleic acids. These range from around 70 to 90 nucleotides, and they have a unique clover leaf structure. If you draw it out in 2 dimensions, you see that there are some areas, there's some sequences in the RNA that are complementary, and they pair up. It's sort of the way nucleic acids do into a double helix, but we don't see a double helix structure in nucleic acids. The 2-dimensional structure is a simplification. You see to the right of that, you see a more accurate 3-dimensional depiction of what this looks like. And two things you see there are, you have what they call an acceptor stem, which binds an amino acid, and then what's called an anticodon, which is specific for a particular amino acid. We will see how that works. The anticodon binds with a codon on messenger RNA and in the process of protein synthesis. We're going to go through the whole sequence of protein synthesis from DNA all the way up to the protein itself. RNA molecules, you know, they're nucleic acids. They're polymers, they're polymers of nucleotides, and they differ from DNA molecules. And let me stop this for a second. I need to correct something here. Okay, there we go. I rearranged a lot of the slides, reordered them, some of them, and I changed some of them quite a bit. And that little DNA thing got through. Okay. I said RNA polymers are polymers of nucleotides, similar to DNA. We've seen the structure. We've seen how they're put together. Here's the differences. The sugar in RNA is ribose. You remember that. Remember DNA, D stands for D, for the D in deoxy, which means it's a ribose molecule, where the 2' hydroxyl is missing or is absent. In ribose, it's there, so it is not a deoxy sugar. It is the ordinary monosaccharide aldopentose, and of course it's bounded to a base, either a pyrimidine or a purine, and then on one end and on the other end to phosphate. Uracil replaces thymine. Remember that. Thymine is in DNA, yet uracil is the equivalent in RNA. So in places where you actually you're going to see this in this section, where if a thymine is present, is the base in a specific site on DNA, on a DNA template, when it gets to the stage where you have the RNA equivalent, it's going to be replaced by uracil. RNA molecules are single-stranded, they're not double-stranded. You just saw on transfer RNA that there is a little bit of complementary base pairing that goes on within an RNA molecule, but RNAs do not form double strands as do the DNA. RNA, if you think about it, the difference in the sugar is itself quite a substantial difference. I don't know if that is the reason, but it is a fact that RNA does not form double helical structures like DNA. RNA molecules are much smaller than DNA molecules. Large RNAs are probably going to run into maybe thousands of nucleotides for like a large protein, but nowhere into the billions of base pairs that can show up on a DNA molecule. DNAs are, just the nucleic acid alone is huge, and we haven't really talked about all the associated proteins, the nucleoproteins that go along with it. So those are pretty much the distinctions between DNA and RNA. And there they are. Now, we're going to introduce we're actually going to introduce the genetic code in the next section. But RNA makes up, well, it transmits the genetic information needed for cell operation. RNA is going to carry with it the sequence of a protein translated. Messenger RNA is going to do that, but it's going to carry the sequence of a protein which it transcribes from the DNA in the form of nucleic acid, triads of nucleotides. So there's going to be 3 letters in each. You'll see this next section, but there's going to be 3 letters to a codon, it's called, and the RNA is going to transfer information from the nucleus to a ribosome, and then the ribosomal RNA is going to get in the act, the transfer RNAs are going to get in the act, they're going to make a protein. RNA is most of the nucleic acid that's found in a cell. Protein synthesis is going to be going on most of the time. And you're not going to be able to do one molecule at a time. You're doing this. There's a lot of protein synthesis going on, so you need a lot of nucleic RNA to take part. There's 3 types of RNA. I already mentioned these. Messenger RNA carries the genetic information, as I said, from the DNA in the nucleus to the ribosomes in the cytoplasm. We'll see what a ribosome is momentarily. Transfer RNA interprets the genetic information in messenger RNA. It recognizes a specific site, a specific codon on the messenger RNA, and the amino acid that corresponds to that codon is going to be attached to the transfer RNA, and it's going to bring it to the ribosome for the synthesis. Okay. Then there's ribosomal RNA, which is the most abundant type. It's combined with proteins to form ribosomes. And here's some statistics on it. How much percentage of the total RNA? You see 80% ribosomal, messenger RNA 5, and then transfer RNA 15. And you'll see, when you see the details of how this is done, you might see how this is kind of a reasonable breakdown percentage-wise. Okay, let's talk about the individual RNA types. We saw the ribosomal RNA is the most abundant, and it combines with proteins to form ribosomes. Now, your text doesn't really say much about ribosomes, and I think a couple words should be said. These are composed, well, the text says these things, but they are composed of proteins combined with RNA. They consist of 2 subunits, a large one and a small one, and they're the site of protein synthesis. Now, the ribosomal RNA it might actually have some itself. There are ribonucleic acids that actually have some sort of a enzymatic property to them. These can be called ribozymes, and these things are going to be a combination. The combination of proteins and nucleic acids is kind of an interesting from an evolutionary standpoint, because there is some discussion over whether life evolved protein first or nucleic acid first. So There's some thought that the original life forms were based on RNA as having the enzymatic activity, and the proteins only took over that responsibility later on. But we don't really need to worry about that here. But that is a little bit of an aside. Messenger RNA, it contains genetic information, and the ribosome graphic is still there, but it's not totally irrelevant because the messenger RNA carries genetic information from the nucleus to the ribosomes, and it serves a protein for a template for protein biosynthesis. So you will see in actually in a future slide the basic process of what goes on in the ribosome in the synthesis of a protein. And that'll actually be more in the next section. So just keep this picture in mind. And transfer RNA, we saw that there's a lot of them. It's a pretty hefty percentage of the total RNA, 15% in a cell. These are small molecules, 70 to 90 nucleotides. There are a lot of them. There's going to be one for each, at least one for each codon in the genetic code, which again we're going to see this in the next section. So these carry the amino acid, each amino acid, to the ribosome for protein synthesis. All right, so they have they're all similar. They have similar shape, the similar if you spread it out in 2 dimensions, you get the clover leaf shape, and if you have it in 3 dimensions, you see what's on the right. They have a 3' end, which has a nucleotide sequence ACC, and it's going to be adenine, cytosine, that's the acceptor stem. That's where the amino acid is bound. There's also an anticodon, which you see labeled in red, which is a series of 3 bases that complements a codon. 3 bases on messenger RNA that corresponds to an amino acid. So whichever, they don't give you the specific, they don't give you the sequence of the anticodon here or here, so we can't really tell specifically what amino acid this is, but we will see transfer RNA in the next section, which has the anticodon labeled, and they haven't lined up with the codon on the messenger RNA. So this will be a little more clear after that section. We're going to say a few words about protein synthesis now, which is a process. There's what's called the central dogma of molecular biology, which says that information proceeds from DNA through RNA through to the protein. So that's the direction of information flow. Watson and Crick, I think it was Watson, actually regretted having called it the central dogma for one thing, because in science really there's in principle no dogma. Anything is subject to further refinement or changes. It could be proven or disproven. And in fact, we're going to see in this section, when we talk about viruses, when we talk about retroviruses, we're actually going to see a case where information goes from RNA into DNA and then back through. So, but it was called a central dogma. We don't really talk about that here. That's just a historical thing that I mentioned. Okay, protein synthesis occurs in a couple steps. Transcription that occurs in the nucleus, where genetic information is transferred from the DNA to messenger RNA. So messenger RNA, when a protein is being synthesized, the first thing that happens is that a messenger RNA is synthesized in the nucleus using a stretch of DNA, which we call the gene, which is this piece of DNA, the section of DNA that codes for that protein. And the messenger RNA makes its way out of the nucleus, and then it gets to, it lands in a ribosome, runs through a ribosome, almost like a tape reader, if you've ever seen one of those. And it's translated into protein by means of transfer RNA molecules that are charged with the appropriate amino acids, as we'll see. And here's more pictorially what happens. You're in the nucleus, you see you're taking the DNA, and a section of DNA is transcribed into messenger RNA. The messenger RNA is being synthesized there, and it makes its way out to the cytosol, which is area outside the nucleus, and then it gets into a ribosome, and there it is. It's translated into a protein molecule, as you see. So this is just actually recapping what I just said. You notice the core chemistry skill. They're going to want you to write a messenger RNA segment for a DNA template. So this is recapitulating here. Transcription begins when a section of DNA containing the gene unwinds, and there's a battery of enzymes. Yes, there are enzymes that actually have to operate on the DNA to kind of untwist the spiral and open up a segment, open up that part of the DNA, break up the double helix, so that the enzymes can get in and make the messenger RNA. And then of course it all comes back together when the messenger RNA leaves. Then you have messenger RNA, as you see there. You're taking the DNA template strand as a template, and then the RNA bases are lining up complementary to the DNA template strand. And notice where you would have a thymine in the DNA, you would have uracil on the RNA. The first one, on the first of those base pairs on the right in the diagram, where you had adenine in DNA, it's going to pair with thymine in DNA, but this is RNA now, and you have uracil instead of thymine. Keep that in mind. And otherwise, it just lines up the way the base pairing works in DNA, as we've talked about. You have a RNA polymerase, which is the machinery essentially that makes the RNA. And you see you notice that what they have here is that the double helix has been unwound to some extent. And I'm going to blow this up so you can see it a little better. The direction of it's giving you the direction of transcription. This is it's going from 5' to 3', but that's really, they don't talk too much about that, the 5' to 3' polymerization, but that is the way it works. You have the RNA polymerase, and the whole assembly there will unwind a little, what they would call a replication bubble or something like that, and it's going to move along until it gets to the end. There's what we call I'm using the terms codon and anticodon. Those are 3 base segments that code for an amino acid, but there's a couple of them that actually act as punctuations, the start codon and a stop codon. So when it gets to the end of it gets to a stop codon, it's just simply going to stop it's going to be the end of the sequence, and it's going to, the DNA polymerase is going to fall off, or the RNA polymerase is going to fall off of the DNA. DNA is going to snap back together, and the the messenger RNA is going to head out into the cytoplasm looking for a ribosome. Okay, now we talked about this is, you recall, the learning goal here. Write the messenger RNA segment for a DNA template. They give you 3 choices here. Okay, this first one here, this first sequence of bases, CTAAGG, is DNA. Okay, and they want you to determine this is in a DNA. This is a DNA template. So what's the sequence of bases going to be in the messenger RNA that's being synthesized? And you really don't even have to think about this one. Now, let's think it through. Well, first I'm going to tell you the secret. There's only one sequence in there that has uracil in the RNA sequence. You notice A and C have thymine in there, and thymine doesn't show up in ribonucleic acid, only in DNA. So B has to be the correct answer if there's a correct answer. But let's think it through anyway. Okay, cytosine, what does it pair with? It pairs with guanine. All right. Thymine pairs with adenine. Okay, the thymine in the DNA is going to correspond to an adenine, so that isn't going to be a problem. So you've got the adenine there. Adenine in the DNA is going to pair with, it's going to thymine in DNA, but it's going to pair with uracil in RNA, and there's 2 of these, so it's GAUU, and then G, the guanine, is going to pair with cytosine. So you see that B is definitely going to be the correct answer. And there you have it. Okay, we're going a little slowly getting a little deeper into protein synthesis. We've seen now the transcription part of it, and we're going to, the next section, it's going to get a little more complicated, but what we're going to see is the genetic code and protein synthesis. So you're going to see the translation part of this transcription translation process, the central dogma, as they called it, in actually coming to its completion, where we're actually going to finish up with a functioning protein molecule when we finish the next section, which is going to be 17.5, the genetic code and protein synthesis. And I will see you then.